So, good afternoon, everyone, and thank you very much for joining this tour rather than the other about the two-way street of citizen science and disease prevention. So I'm the first speaker, and I will start right over. I'm Gábor Földvári. I'm working at the HUN-REN Center for Ecological Research. And you can see the title of my talk. I will try to take you to a tour which is about how we can prevent emerging infectious diseases with the help of citizen science and the DAMA protocol. First of all, I'm really grateful to the funding agencies that are helping my research group to work. One is National Laboratory for Health Security, the other one is the Tripartite European Horizon Action, and the third one is a Coast Action, which I'm happy to chair. I am coordinating over 280 scientists from more than 30 countries who are all working on prevention and anticipation of ticks and tick-borne pathogens. And luckily, it will continue this year because I got another grant from the National Research Excellence Program, so we are happy to continue our work. And this is the research group that I am leading. It is not by chance, or maybe just a bit of chance, that we are half women and half men, and I'm really honored and happy to have also Dan in my team, and mentors like Lajos Rózsa, and we have BSc students, master students, PhD students, and postdoc, so it's quite well-balanced team of scientists. We are working on the ecology and epidemiology of pathogens, and first of all I would like to introduce to you the so-called parasite paradox. This means that you know pathogens and parasites as ecological specialists. This means that they are usually within the same hosts for all their life and for during evolutionary times. However, on the other hand, we can observe very often that we have many cases of disease emergence, many cases when pathogens are jumping from one host to another. Why is this a paradox? Because we had a wrong theory, actually. So the classical so-called co-evolutionary model tells us that for a pathogen, in order to switch to a new host, it needs to be genetically modified or changed. There needs to be a mutation that enables the pathogen to jump to another host. By the way, the slides are from Orsolya, and she agreed to use these. Oh, sorry. That's already late, yes. So if it's true, then based on this new mutation, we can say that emerging infectious diseases are rare events and they are unpredictable. So we have a good and a bad news. The bad news is that they are not rare, as you know, since we had COVID, for example, and many others. The good news is that they are not unpredictable either. Why not? Because we have a much better functional paradigm. It is called the Stockholm paradigm, and then Brooks and colleagues described it in the last 20 years that actually there is a huge variety, both genetic and phonetic variety of pathogens within, for example, a bat host, a wild-living mammal, and some of them are able to switch to a new host if there is opportunity. So the key here is to have an opportunity to jump over. And as we experienced during COVID-19, there were variants which were able to infect dogs and deer and humans and rodents. And we can say it's frequent and predictable, and within the new host, as we again experienced with COVID-19, the new variants are already able to spread more efficiently compared to the original one. So this is all described in the 2019 book. So this book appeared just before COVID-19 entered our small world, and it is based on the so-called ecological fitting, which is a very important observation from the 80s from plant-feeding insects, that there is no need for a genetic change for a pathogen to switch to a host. Only opportunity will lead this pathogen to change, and opportunity means climate change or habitat loss, for example, which leads to the migration of mammals and other vertebrates, of vectors like mosquitoes and ticks, and of pathogens. So the more opportunities, the more host switches will occur, and this is why we can say that emerging infectious diseases are much more a rule than an exception. As all of you know, climate change affects everything and everyone, and not only the global global temperature increase, but also these sudden and very drastic climate events or weather events For example, in 2024 in Valencia, within 15 minutes, this huge flood came over to the city and over 200 people died. And this kind of events, of course, change a lot in the habitats of the vertebrate hosts and also the vectors and the pathogens. With the network analysis, scientists also showed that the different factors of global warming and climate change, like warming itself, precipitation, floods, and so on, is affecting the most vector-borne diseases, but also waterborne diseases, airborne, and so on. And to the right, you can see the bigger the letter, the more bigger the effect of climate change. So we can say that especially for viruses, bacteria, and parasites, climate change is exacerbating the spread of these diseases. Another factor is also very important, the destruction of natural habitats. If we decrease the biodiversity, it will again create new and new opportunities for pathogens to spread. With a very simple metaphor, if you shake a tree, something will fall down. This is what happens if you build roads, if you extend cities, and so on. Another problem that we only know a very tiny vicinity of pathogens and microbes around us. So we know, for example, around a couple of thousands of mammal and bird viruses, and according to the estimates, they are something like 1.7 million on the globe. This means that around 500,000 of these viruses are able to infect humans, but we only know a very, very tiny fraction. And if you understand more the money language, we can see that BC, so before COVID, we have been spending around 1.3 trillion US dollars only for other emerging infectious diseases to treat and to respond to these emerging infectious diseases. And just a single EID has cost us already several tens of trillions of US dollars. Another factor which is affecting emerging of pathogens is urbanization. As you know, more than half of humanity now lives in urban big cities, and it will be very soon 2/3 of humanity, which is not actually a safe place for being protected from pathogens. Think about these green spots in the cities. These are very important recreational spots for humans, but also safe havens for rats, for hedgehogs in Europe, or for ticks and mosquitoes, which are leading to these places to be hotspots for some disease emergencies. And if you go to Southeast Asia, on these wet markets, you can see the perfect storm, because we have dead animals, we have living animals, we have humans, and we have many, many microbes around. So this is the place where the real host switching or the real emergence of disease happens. And if you add another amplifier, which is globalization itself, then you can imagine that anytime anything can come out. So as in one of Dan's papers I read, we live in a minefield of evolutionary accidents waiting to happen, and this is not just a metaphor. We can put a real map with real emerging infectious diseases, and this is a terribly old map. It is coming from 2017. We could do triple or 4 times more pathogens on the map now. So after this, lots of bad news, the question arises, what can we do as scientists or as people interested in this topic? The good news is that based on the Stockholm paradigm, we can be actually proactive about coping with the emerging infectious diseases, because we know that colonization of a new host requires only new opportunities given specific pre-existing capacities, and because these pre-existing capacities are predictable, they allow us to anticipate the disease emergence. Not 100%, but we can anticipate the emergence of diseases. And a very practical method to do this is the so-called DAMA protocol. This is an acronym for documentation, assessment, monitor, and act. I will quickly show you how it works. Very easily put, it's also called finding the pathogens before they find us. So documentation, as I mentioned, we know only a fraction of pathogens and microbes around us, so we have to document to find what is around. And if we find something, of course, we cannot monitor everything, because we have limited capacities, resources. We have to assess what kind of threat does it mean to humans or domesticated animals. This is a threat or a risk assessment. And then we go back and monitor. We have to check what is the timely change, what is the special change of these pathogens. And this needs lots of money, lots of time, and lots of efforts for many researchers. And the last one is very often missing in scientific research projects, to act, to do something to prevent the next outbreak. So documentation can be done by researchers, can be done by citizen scientists. A very simple way of what I'm doing here is not in a war zone with the white flag. It is the method, it's called flagging, how you can collect ticks, blood-sucking small parasites within a forest, because they are attaching to this flag, and then you can count them, take to the lab, and do your further experiments. So the next step, after you have documented something, you have to assess the risk. This is the laboratory work. You check, for example, for the DNA of the pathogens in the molecular lab and find out whether it is risky or not. So the assessment is also called a phylogenetic triage. You may know the word triage from the medical literature, when during war times the physicians had to make categories of which wounds are very severe and which are not so severe, so the people can fight over. So in this case we do this triage based on the phylogeny, on the DNA of the parasite, which means we put the question, is it a known pathogen or a relative of a known pathogen? If the answer is no for both questions, we archive it and don't work anymore with this right now. But if the answer is yes for either of these questions, we have to check more information. How can we find this? What are the threats if we find this pathogen in a given environment? And the next step, as I mentioned, is monitoring. We have to go back, check the temporal and spatial dynamics of the pathogens. This is a very time-consuming part. And as I mentioned, action is a crucial one. And we scientists are very good at describing, so documenting, very good at assessing, and very good at monitoring, but we are failing usually in this act part. This is one of my favorite slides about citizen science, and now I want to turn to the point of citizen science used as a kind of action for preventing diseases, because there are many people who want to contribute. They are really willing to do something, and we scientists have lots of tools, lots of questions, lots of tasks. We just have to connect these parts somehow. So for this reason, we started a citizen science project finding a tick species, or actually 2 tick species within Central Europe that are not endemic here, but they are carried by migratory birds. These are called hyaloma ticks. These are quite big, quick-running ticks, and they usually feed as juveniles on migrating birds. I think it's not working. So the migrating birds take the larvae and the nymphs, and they can be carried for several thousands of kilometers from Africa or for the Mediterranean countries to Central and even to Northern Europe, even in Sweden. And they drop off from the bird, and they will look for hosts like humans or cattle or horses here in Central and Northern Europe. The problem is, it's not only interesting for tick biologists, the problem is that they are able to spread Crimean Congo hemorrhagic fever virus. These are the juveniles, and this is the virus itself. As the name tells us, it is a quite debilitating disease. There is no vaccine, there is no specific treatment, and it is, according to the physicians who were working with patients of CCHF, told that it is looking worse than Ebola, actually, because if someone reaches that point that there will be a hemorrhage, then more than 30% of people can die in this disease. So it's not only a biological interesting question, but a real public health effort to find these ticks and to find the virus itself. So that's why we started with a student, actually, a website and a citizen science project called Tick Watcher. This is the Hungarian logo for it. And we were explaining, first of all, what kind of ticks do they have to look for. So here in the middle, you can see the common sheep tick, which is the most common all over Europe. This is transmitting Lyme disease, Borrelia, or tick-borne encephalitis. This one is very common on dogs, but hyaloma ticks are the double of the size of these, and they are very quickly running, and they can even be observed or differentiated by naked eyes because they have stripes on the legs, as you can see. And we explained what they have to look for, and we asked people to make photos, send the photos to us, and also send the ticks to us for checking. And here to the right, I think I will need some help with the video. Kis segítséget kérhetek a videó indítás, köszönöm. So you can see what I mean by quick running. This is normal. This is the normal video. No change. Yes, so I did it myself in Turkey. Yes, and actually, you don't catch them; they catch you. But that's true. So usually I am hunting the ticks, but these are called hunting ticks. They are running like hell, and they can perceive. If you clap, for example, they can perceive when they run after you, like for several tens of meters, and really scary, really scary. And the bad news is that we have also already in Hungary these guys, yes. So we are lucky actually that it's so terrible, quick and so scary because people pay more attention. So this is good for us. Of course, if you manage to make a good website, it's not enough because if people don't know about it, then it's not working. So we went to every kind of public outreach opportunities. We showed how ticks look like. We had these plastic ticks and tick toys. And at every conference, I take this, these toys from ticks. And people are very interested because on one hand, they are afraid of ticks, but on the other hand, they are interested. How can they prevent the bites? What can they do against it? So my team is very enthusiastic. My PhD, yeah, someone is not so enthusiastic. But my PhD student even made an escape room with the topic of ticks, and it's a huge success. We did it not only in Hungary but also in Serbia during the Researchers' Night, European Researchers' Night event. It was a great success. So this is the way how we get to people, how they are getting interested in ticks, and then they start sending in photos. First, just bugs, and beetles and spiders. But after a while, we received, of course, ticks. This citizen science project runs for 5 years now, so we have pretty much nice photos as well. You have to be very careful with the media. So I quite often give interviews and television, radio interviews. But if they put the wrong photo, like here, then people start sending in this kind of ticks, this species of ticks, which is, of course, interesting as a bycatch, but we are interested in the hyaloma ticks. So this is, for example, not a hyaloma tick. Very nice photos. This was shot by a mother from her daughter's eyelid. This is the common sheep tick, very, very often find either in the eyelid, even in the ears, everywhere on the body. And sometimes not the best quality photos are the most important because here you can see it's a blurry photo, but you can see the stripes. So this is one of the first hyaloma catches with this tick watcher program in Hungary. This was from a cattle, and it was a hyaloma rufipes, also transmitting CCHF virus. We were checking genetically to which clade is it closer, and we found out here that it is the European African clade, so probably this tick was taken from Africa by a migratory bird to us. And a hyaloma marginatum from a dog. Also, we checked this probably came from the Asian side because birds you have also migratory routes from Asia to Europe. Donkeys, horses are affected. Someone found it on the kitchen floor. So lots of interesting observations that we got during these 5 years. We also summarized the bycatch. You can see that in the first year of the study, only these 2 points were hyaloma. Can you check this one? This 15 kilometers from here. This was the first hyaloma found on a dog within a garden, very close to Kőszeg, actually. So just pay attention. And all the others are also interesting because we don't have the scientists and don't have the money to go to every corner of the country. So citizen science can contribute also this geographical extension of our sample size. We also were very interested. So here you can see the summary of the 5 years hyaloma catches. In total, we have now 28 specimens caught. And very interestingly, there are some places like this one here where there is an aggregated number, which means that these are not taken by chance, but much more probably it is a starting local population. So we can expect that during global warming and with the years, these species will be part of the local fauna. So they will be endemic in Hungary as well. And this is the reason why we have to monitor them also for the virus itself. So this is the summary, where and what we found from hyaloma. And we have also received many other hundreds of ticks from other species as well. Okay, this is a quite messy map, but what I want to show you with this, that we are also running serological studies. So we are looking for the antibodies against the Crimean Congo hemorrhagic fever virus in cattle and sheep and in wild living mammals. So here in these places, you can see where there were positive animals, and these are exactly the places where hyaloma ticks were also reported by the public. So this means that there is something going on also with the virus circulation. We were checking all the ticks that we could receive from the public for this virus. The good news is they were all negative for CCHF. But we have found a very close relative of this virus, which has only a name actually and nothing else. It was detected in Russia first, and we were the first who published the full genome of this virus, and we know nothing about this, but what we know is this is closely related to CCHF, which means that we have to be very careful with that. We have to go back and monitor this virus in the specimens. Okay, and as we can read in the Stockholm Paradigm book, our protocols must be also evolvable because there is no static solution for a problem which involves an evolving Earth and evolving biosphere. So for this reason, we also went a step further, and last year we launched a mobile application called Pragmatic. And this is running now in Hungary for nearly a year now, and it's also available in English language. And you can not only make photos and send in, but also the location is possible to record, and of course you can send in the specimens as well. So here on this map you can see that in the first half year we have as much observations as during 1 year with the website. So the mobile app is really working well. So we really hope that it will increase our efforts to monitor the presence of the tick and also the virus. And one more important thing that, as I mentioned, I am heading this European consortium on ticks and tick-borne pathogens. So I'm also happy to lead the 4th working group, which is especially about citizen science involvement and the DAMA protocol. And since we have more than 30 countries involved, I was asking all the colleagues that please translate it to your language. So now it is being translated into over 30 languages. It will start this year, hopefully, in most of Europe. We have even some exotic places other than Europe. So as it will be translated, it will be also available for people living in France or Spain or elsewhere to send in ticks to the local colleagues, and they will check what ticks they detected. So I really hope that this will be functional. You can also download it here. As I mentioned, 15 kilometers away, people already found hyaloma ticks, so you can check also tomorrow in the coffee break whether you have ticks. It's already tick season because it's springy weather. So I recommend to use it, and I really hope that by this or with the help of this tool we will be more and more proactive and apply the DAMA protocol as far as we could. Thank you very much for your attention.
Okay, hi everyone. My name is Orsi. I am from the Medical University of Vienna, and I am a very long-time friend and colleague of Dan and Gábor, both. So what I'm going to be focusing on more, and this is where I started working first in Austria and then at the Medical University, is I was very much interested in this action. So I want to touch up on Gábor saying that scientists, I'm an evolutionary biologist by training, right? So I did documentation, I did assessment, I did monitoring, I know what that was, but then when action came to be, we were kind of a little lost. And so I currently do viral surveillance for non-zoonotic diseases. And so I just want to give you a little bit of background on where I think that can make a huge difference in how we approach emerging infectious diseases, but also diseases in general, right? Known infections. So this being the UNESCO conference, I'm sure everybody's familiar with the SDGs, right? The Sustainability Development Goals, which is a very ambitious target, right, to reach. But nonetheless, right, we are currently at the midpoint, more or less, of 2030 targets, and 15% of all the targets are the ones that are on track. This is according to the last report in 2024. And so what I think is crucial here is that most of the reasons as to why they're off track is missing, skewed, and non-representative data. It's the data that we don't have. A lot of these, we don't know if they're off track. We just can't prove that they are on track because we don't have the data. And of these, what I was particularly interested in is this was recently published at the end of 2024, is that an alternative to how we approach and collect and analyze data from epidemiology and disease and anything in general could be the citizen data, is what we call. So they published this framework on citizen data collection, which this article was about, and it really fed into what we were doing. So our key interest was SDG number 3, which is good health and well-being, and within that was the infectious disease framework. Just to give you a few numbers as to why we're not exactly on track. So COVID-19 reversed progress to levels of 2005. The share of communicable diseases went from 18% to 28% from 2019 to 2021. It's not doing much better today. HPV vaccine coverage was 15% in 2022. I'm sure some of you have seen the targets of WHO, which was getting 90% of girls vaccinated. This is not happening yet, right? And again, it was the data, right, that was missing. So this report calls for prioritizing prevention, but that faces data gaps and distrust. And so this is especially expressed in access to high-risk populations. And by high-risk populations, we mean populations who are either at a higher infection risk, have a higher prevalence, a higher pathogen diversity that are not represented in this data because we don't have access to them. And this was really kind of the place we were coming from. So we said, okay, let's see why they're not represented. So when you look at infectious disease monitoring, and this is the lab that I work in does, right? We monitor infectious diseases, we monitor viruses in particular, but it's very similar for bacterial pathogens. Infectious disease monitoring has 2 main data streams coming into it. One is patient data. It's stuff that comes out of hospitals, and the other one is environmental surveillance, wastewater-based epidemiology, airborne epidemiology, you name it. So what do each of these do, right? So patient data is wonderful because you get a genetic aspect to it, you get the pathogen itself, and you also get social measures. However, patient data is going to be extremely skewed because for someone to show up in the database, you need to have health insurance, right? I'm looking at my American friends, right? You need to have access to healthcare, and you need to choose to seek healthcare, right? So if I say, COVID home officing for cab drivers who do have health insurance, and they may have access to healthcare, but they don't have an option to stay home because then they're not making any money, right? So these are filters that people have to go through to show up in this database. And then of course physicians would be nice if they uploaded stuff into the database, but it's a lot of loops to jump through before you're in there. Now on the other hand, you have environmental surveillance, which is great. Because wastewater, we can sample freely, we don't need consent, which is something science often really likes. If we don't need consent from anyone, I can just take what they dropped, literally. So it gets a very nice coverage of the population, but there is no social dimension to it. I do not know whose sample I'm collecting. I don't know if they live in the area, I don't know if they were transients, I don't know if they're travelers, I don't know if they're commuters, I know nothing about what's up there, right? And so to make matters a little worse, you have what we would call high-risk communities, right? These are communities either along socio-demographic angles or geographic angles or you name it, some sort of angle that are interconnected with one another. They're for some reason disproportionately affected by infections, so they have a higher prevalence rate. They often cluster around a demographic, and they show high pathogen abundance and diversity. All of these things they do, unfortunately what they don't do is show up in either of the databases, right? And if they do, we don't know if they do. And so we said, okay, what can we do? Right? What is this? How does this change the way we understand data? Well, first of all, epidemiology doesn't get data out of these communities, so we don't know how the targets are doing with them. But another crucial thing that I think COVID-19 taught the world that maybe some of us had understood before is that epidemiology is not having impact here, right? These are typically marginalized, underrepresented groups who are disconnected from the scientific message, from the health authorities, from the government, and they're not willing, they're not able, they don't have the capacity to comply with prevention measures, which puts them at even more risk. And so within this data and impact, we said, okay, what do we need to change this? And we said, oh, well, we need two things for data. We need molecular and social data, that is for sure. We can't go with only one, only the other. There are a lot of methods that can collect one or the other. We need this together, right? We need to understand the connection here. And on the other hand, we need involvement. We need the community to be there and do this with us. We can't just trot in, do our science, trot back out, because that's going to leave us without impact, which is exactly the case that the issue we're facing right now. And so in terms of what we need, we said, okay, where is this expertise going to come from, right? Molecular and social data is going to come mostly from science, right? Because we're used to generating genetic and all sorts of different social and whatever types of databases. When it comes to working with the community, we need to have people involved who are local, who have what's called lived experience, right? Who can tell us about the contextualization of the issue. And we also need to do involvement in a safe manner, which means we need another kind of expertise here, which is the legal policy-based expertise. And so if you look at this, infectious disease monitoring is actually an issue that requires a lot of different expertise other than science. And as it turns out, apart from these, there's another layer, the strategic insight, which is gonna come from different kind of societal stakeholders, right? So with all of these identified, we said, this is fine, but how do we get all of these things together? And this is when we said, okay, let's look into these so-called multi-stakeholder programs, right? And let's translate all of these different people into a framework that we can understand. So we have what's called local case actors, right? It's the local people who are suffering from the transmission, who exhibit the risk factors, who the health policy is implemented upon. These are citizens, patients, communities, you name it, right? The lived experience. The practice experts here are going to be the ones who understand healthcare policy, the authorities, they understand the legal environment, the policy environment, what can and cannot be done, how the interventions can be planned. Science over here is going to be represented by medical research, epidemiological research, all sorts of treatment plans, and then the strategic case actors, who are the ones who deliver the service and the healthcare system. This is going to be pharmaceutical companies, biotech, NGOs, hospital staff, who are somewhere in between the policy environment and delivering it to the community, but not members of the community, right? And also not active researchers. And so all of this led us to frameworks where you can identify societal problems and scientific questions, right? The societal problems are going to be dealt with the local and the strategic case actors, and it's going to result in some sort of practice. This is going to be governed and overseen by practice experts, and then the scientific questions are going to be looked at by scientific partners and lead to some sort of scientific advancements. But as you see, this is two different sides of the page, right? This is what's been going on during COVID, during a lot of different disease managements that were not super successful. So these frameworks, Jan et al. In particular from 2012 was what we applied here that says, okay, what you can do in this case with this empty middle area is fill it with three things. One is framing. Identify topics of mutual interest to society and science, right? Not just what's publishable in high-impact journals, and not only stuff that leads to, you know, a better tomorrow, but something that is of mutual interest. When you have mapped some overlap in the motivation, you can go to what's called co-creation. You develop new concepts and solutions together, right? And the emphasis is on this part of the word, right? It's co-creation. You have local people, you have stakeholders join in on your workshops, on your brainstorming, on your data collection, on your analysis, on everything you do. You let them guide certain parts of the issue. And then, there we go. And then you implement your findings. And you see how that implementation works, you test it, you do some feedback, and hopefully this is going to contribute to both the practice and the scientific advancement. So this was the theory that we thought, okay, this is worth a try. We can give this a go. So I'm going to present to you two case studies, two projects that I'm currently working on. This one is funded by the FWF, which is the NSF for Austria, essentially, right? The Austrian Research Fund. And this is called LoopFees. So this was done on respiratory pathogens. Our interest was pathogen distribution and risk factors for infection. This is not something that we have a very good oversight on, how respiratory pathogens and what kind of diversity we have in Vienna, but it's also of global interest. The societal problems is high infection risk and low prevention for a lot of the groups. Now, the risk group, one of the main risk groups for these kinds of things are students, right? Students in public educational settings, kindergarten to high school. Because of, we can imagine, we've seen students many times, and with the students, teachers who are exposed to the students and spend time with them, right? So they were our risk groups that we contacted. And then we also formed a collaboration, this is us, with two organizations in Vienna who are educational experts. They don't do research themselves, but they do scientific workshops and scientific seminars for students that they can participate on for a nominal fee or free of charge. They have their own lab spaces, they have their networks. And so in terms of the framing, we said, what's of mutual interest to us and to the schools? It's what kind of pathogens are there in the school circulating among students? This is something the school is interested in and something that we were interested in. We then took this opportunity and developed an infectious disease monitoring program with the schools and the students that was run exclusively by the students. We were not physically there. I'll show you the flowchart. And then once we acquired results, we channeled those back to the schools, and with the help of the schools, they got channeled back into the curriculum of the school. So students were actually learning about the results in class and about the wider scientific applications. And with this, it's not just about a better understanding of how viruses work, but what we really kind of honed in on was ownership of students, right? That they had agency in the program. They understood the scientific process of, this is what I did, this is how I came up with it, and that leads to something, and I can actually do this with other things, right? And then for us, we were able to conduct epidemiological analyses, and we were able to apply transdisciplinarity, or TD, into infectious disease research, which has not really been done before. And then we had the Federal Ministry of Education kind of overseeing the project, because they're minors, right? So all data is sensitive data here. And so this is the flow chart of what we had done. This is out of the article, so it's eye candy for now. But what I do want you to focus on is that in the steps of the study we had done, leading to like 3 different kinds of readouts and data analysis tools, here is the color codes for the different stakeholders, right? It would be researchers, teachers, and students. The frames are going to tell you who had participated in each of the study sections or study steps. And over here, molecular sampling, there's no purple for researchers. That was run by the students, as was social sampling. There were parts, lab analysis, where it was teachers were absent. It was exclusively students working with researchers. Same goes for statistical analysis, right? So we just really let the steering wheel out of our hands and allowed them to do it. And this was really a pilot test to see if the data that comes out of it is worth something, right? So all in all, we worked with 6 different schools in Vienna and Lower Austria. We had 8 teachers, 47 students involved. This is, you see the student count per, the participant count for each of the schools, and then the heat map shows you the student to teacher ratio, right? So sometimes there was one teacher, 2 students, sometimes there was one teacher and 19 students, right? Involved in different schools. And then what we got out of it, and I'm just gonna summarize it, so if someone's interested, please reach out to me. So the molecular readouts were absolutely robust. So much so that we could process them in the same pipelines we process wastewater in, we process hospital data in, and they were actually directly comparable to one another. So you could mirror the relative abundances that you get out of hospitalized cases with those that we get out of schools and see the differences. We did sequencing on the samples, so the sequence data was also something that we used to kind of do this multiplex detection. We compared abundance and diversity against CO2 readings, so we were able to match that up with environmental readouts. In terms of the social attitudes, we were able to test different prevention measures and look at which of those are the ones that students believe are efficient, but they face what's called a feasibility barrier, so they cannot comply, and which of the ones are the ones that students think don't work at all, even though it's very accessible to them, right? So we were able to distinguish these things, and we were also able to use network analysis to map the entire design by participation rate in different, so this alluvial shows you the participation rate. This shows you the static network of stakeholders with the edges showing you the different interaction networks that changed in different parts of the study. So you can really track the progress of how these stakeholders are interacting and what exactly that leads to. And though this is right now under review in Nature Sustainability, and over here, this is the screenshot of the manuscript. The yellow is gonna show you the students who are co-authors on the paper, and the green are the teachers who are the co-authors, right? And there's me and a couple of other insignificant people, such as myself. But it's the students and the teachers are there, right? That's what matters. And then this is case study number 2. This is HP Vienna. Now that you know the framework, this is on HPV. Scientific questions, HPV diversity in Vienna, or globally for that matter. Just in comparison, so GenBank has about 18 to 20 million uploads on SARS-CoV-2 and 30,000 on HPV, right? Not a very good ratio of how much we understand genetic diversity and the societal risk factors. The societal problems are that there is little to no access to cancer prevention for some risk groups in Vienna, and there are huge issues with implementing any kind of HPV policy. The experts here in this for the local case actors are people without health insurance in Austria who do not have access to any healthcare treatment or healthcare services. And the only clinic, this is a charity clinic working with these people who provide some sort of services. They are the strategic case actors. So they were the ones that we reached out to develop this study. And this is a joint study between us, we're doing the genomic epidemiology, and then by us I mean we're also doing the transdisciplinary framework, because that's me, that's the one-man army. We also have the Univeen involved. They're the ones responsible for the social analysis, and the TUVeen is doing the epidemiological modeling on this. And so the framing was HPV transmission and mitigation, right? That's of mutual interest, 1 for the population and 2 for science. We developed an HPV infection testing system for the people without health insurance, which is free of charge for everybody. And we also asked them to complete a 15-minute social survey to gather both genetic and social data. And this is to be implemented into a policy blueprint that is then going to be submitted to health authorities in an effort to improve access and implementation. And of course, you know, science is going to get its piece too, because we would like to publish this, but what really matters here to me is this part of it, right? That like is tangible. So in this project, this is overseen by the Federal Ministry of Health, and we are here right now. This started last year, so we are very much in the co-creation part that I very much enjoy. And so all in all, there we are. So to kind of get some conclusions, right? So transdisciplinary frameworks for epidemiological surveillance was able to provide us with molecular and social data to fill in the data gaps, but also with involvement to increase the impact a little bit. And so, okay. So in terms of what we now call transdisciplinary epidemiology, right, which is a scalable epidemiological surveillance that is not replacing but complementary to what we call the traditional epidemiological surveillance. If you want to look at all the different data sources epidemiology uses and all the different data types that we collect, right, environmental, molecular, social, there are some who collect very high quality data, but it's non-representative for high-risk populations. There are some who are very representative for the high-risk populations, but data quality isn't the best. And we think, of course, but we are kind of trying to show that the transdisciplinary epidemiology not only delivers all three data types, but it's a nice quartile of this graph to fill, right? With very good data that we can test in many different ways, very good coverage, and high representation. So on the one hand, right, we can deliver data parallel through targeted sampling, all different data types, and, and for the impact, we have practical outcomes for the societal partners. We have focused analyses, which improves representation, co-development for contextual integration and involvement, just to improve ownership, agency, and trust. And so this is just to give a thank you. This is our lab. This is a lab of socioepidemiologists we work with. Those are the people from the schools project. These are the people from the HP Vienna project, right? And so I guess what we're all trying to say here is that changing the way we look at epidemiological data and data collection is good, right? It's not about improving the things we do now. It's about doing new things and changing into new perspectives in a way of citizen science being a two-faced friend, right? So thank you very much for...
Well, clearly, what you need to do if you want to be successful is to find some really smart young people and then convince them to claim that they actually learned something from you. Closer to my mouth, okay. This is the first opportunity I've had in 10 years at iASK to actually talk about what I really do as a field biologist in the tropics, and how I ended up, in a sense, doing what I'm doing. Because my recognition, along with Walter Biegers and Eric Hoberg's, Walter working in the Amazon and Eric working in the Arctic, all three of us were involved in basic biodiversity projects. I was doing an inventory of all of the eukaryotic parasites in 1,000 species of vertebrates in a conservation area in Costa Rica. And Eric was working in the Arctic, Walter in the Amazon, and we were all finding exactly the same patterns that medical doctors and veterinarians were calling the emerging disease crisis. And we were finding it in these large-scale biodiversity studies of non-human, non-veterinary hosts. And that's what got us involved in this whole larger area of, or more important area, let's say, of the emerging disease crisis. So I'm going to talk to you just very briefly about that. And let me see if I can do this without, oh, look at that. All right, this is the sign at the Área de Conservación Guanacaste in northwestern Costa Rica. And there are two things you need to know about this. The first is that the sign says welcome. And then it says, this is a World Heritage Site. Now here are two things that you do not see because they do not exist there. There are no fences and there are no guards. It's the only World Heritage Site for natural history whose integrity is maintained completely by the goodwill of the neighbors. And this is a map of the conservation area. Everything that's in green and blue is this conservation area. It's big. This was the brainchild of Dan Jansen. Now, it includes cloud forests, which are super saturated habitats at the mountaintops in the tropics where it's always wet. It's always raining. And it's kind of cool. And Patia here, that word Patia, that's the name of this particular field station. There are also rainforests, tropical rainforests, what a lot of people call jungles. In the background, you see here the volcano, Rincon de la Vieja volcano, which is, as you can see, a little bit of smoke coming up. It's described as a mostly dormant volcano. And the rainforest is wet all the time, and there's a lot of water running through it, and it's very beautiful. And then this very special kind of habitat called tropical dry forests, which are basically deciduous forests where you have a very pronounced dry season where all the leaves fall off of the trees, and a very pronounced wet season where everything, all the vegetation just explodes, and the animals that depend on the vegetation explode as well. So you get these three very different kinds of habitats all interconnected in one conservation area. And what that means is, among other things, it means that if conditions get bad in one habitat type, the plants and animals that live there can move next door and survive. And that's a critical element of evolutionary survival in general under climate change. But it's also the mechanism that provides the opportunity for hosts and pathogens to mix and match. So for hosts to come into contact with pathogens they're susceptible to that they would not have seen ordinarily. So that's where we started seeing all of this host switching. There's some interesting animals that live in the dry forest. Of course, every tropical field biologist has to show a picture of a monkey. The fact that he looks more intelligent than people like me is just merely an accident. There are some amazing things. This looks like thorns on a tree, but these are aphids. There's a bunch of insects. And they tend to be not only really weird and amazing, but they're also really beautiful. I mean, this is why people like me fall in love with tropical biology. Every time you go out in the field, you're going to see something you've never seen before. You have an entire life of always being guaranteed of learning something new. And we also have coastal and marine habitats. This is a mangrove forest along the coast, and sort of beach line and fishing village kinds of habitats. Now, along the coastline, you also have these interesting things like this iguana. Now, you will read in the literature that these big iguanas are herbivores, that they eat vegetation, and that's true. But the question is, why is this thing orange? Okay, it turns out that orange pigment in things like reptiles comes from eating a diet full of carotenoids. Okay? And what that tells you is that these things aren't just eating plants. They're also eating crustaceans, crabs and things like that along the beach. So they cheat a lot. And a lot of the stories that you read in textbooks about things like dietary specialization and stuff like that, go to the tropics and then throw the textbooks out. Now, this area also is the site of a lot of very interesting cultural material. This happens to be a sign along a river that is not, this is not some sort of representation of this is our territory, stay out. This is actually a map saying downstream there are some rapids and some waterfalls. Be careful when you're traveling on the river. Okay, Now, the world that I live in is the following. If you don't like the answers, question your premises. We don't like the answers. We are failing miserably with respect to biodiversity. We're failing miserably with respect to emerging diseases. Here is the fundamental perspective of the Costa Ricans, and this is the forest and water are the source of life and development. And our assumption is, or was, that is twofold. First of all, that local people, if you give them a reason, they will preserve their biodiversity. And that reason only has to be, if you do something with the biodiversity that gives a better future for your children, you will do it. Now, I'm talking about the 90% of the world where people can't afford to do volunteer citizen science projects because they're working all the time. Oops. Okay. Now, just like the emerging disease crisis, the biodiversity crisis is a crisis of ignorance. We don't know very much about the world's biodiversity. And you cannot properly manage things that you don't know about. You don't know it's there. You can't do anything about it. You're blindfolded. You're just messing around. And yet, humanity requires the biosphere in order to live, to survive. And the idea that we came up with in Costa Rica was to professionalize citizen science, to actually give people who had a high aptitude for the kind of work that we needed a real permanent professional status and economic stake in those kinds of positions. And so the salaries that these people that we came to call parataxonomists were initially set at 3 times the national average salary. This was not only to give those people an economic stability, but it was also to let their neighbors know that what they were doing was important. Okay. And their job is to assist foreign and domestic scientists, so think paralegal or paramedic, something like that, in their research, as well as to educate their neighbors and augment basic educational programs. So the people who live in the villages where there are parataxonomists, if they read something weird on a YouTube channel, they go to their neighbors who are parataxonomists and say, is this legitimate or is this just BS? So they're important members of their community. It also turns out that in that one conservation area, 2/3 of the parataxonomists are women. This means a second income in the family. It means economic independence for women. And it means a really good example of what Sean was talking about yesterday. Because the first thing those parataxonomists do is build a house next to their own house for the parents, for the grandparents, so they can watch the children while both parents are working. The next thing they do is at the end of each year, if they have money left over, all of the parataxonomists in one village get together and they pool the money and they provide a salary for an extra teacher in the school. Instantly, the people in the village realize that this is important and positive. Now, where do we find these parataxonomists? Just wandering around, strangely enough. You go into, houses up in the cloud forest, houses down in the rainforest, houses down on the coastal area. And what we did was very deceptively simple. You go into a village and you have a bunch of plant presses with species of different plants unsorted. And you bring in cigar boxes full of insects that are unsorted. And you say, anybody who wants to be a parataxonomist, classify these things, sort them out. And what we discovered was that in most of these villages, there were always 1 or 2 or 3 people who were sorting these things out, as well as the Smithsonian specialists that we had along to vet their results. Okay? So these are people who really know their local biodiversity. They don't know anything about evolutionary theory. They don't care about evolutionary theory, but they know their own location. And that makes them invaluable because in terms of their local location, they know more than any of the foreign specialists like me who are coming in there. And so this is Patrona Rios, and you see here that she's pinning out insects. And she's classifying them and she's preparing them in a way that all the specialists, insect specialists at the Smithsonian will accept all of their material for their own research. They do it for parasites as well. This is a group of parataxonomists getting into the intestines of a crocodile, looking for parasites. And the thing that's interesting for these guys is that as far as the parasites are concerned, none of these people ever even knew that there were parasites in the animals in the conservation area. And as soon as we had a workshop and started showing them what was inside those animals, you can see they just went crazy. It's like, this is a whole new universe for us in our backyard that we never even knew about. This is the husband of the woman I just showed you in the previous slide. And what that produces is graduate students, researchers, parataxonomists, and the Costa Rican management people of the conservation area all working together, all having a good time, and all contributing at a very high level. Contributing to things like the local educational program. So this is called the educational unit for the dry forest. Bring the kids from the local schools in. Okay, now the kids come in from local schools, and who's giving the presentation on what's there in their own forest? It's not some foreigner who speaks bad Spanish, it's their own neighbors. And here's Callie and Patrona, younger. This is the woman who is sorting the insects, and her husband. They're up in Patia in the cloud forest with their 2 kids. Turns out that one of the byproducts of this program is, in 1 generation, a massive drop in fertility rates. They had 2 kids, both of whom have gone to university. Patrona said that one of her earliest memories was when she was about 3 or 4 years old, sitting on the dirt floor of her house, looking up at her mother, who was holding a baby, pregnant, and stirring a pot. And she said, I said to myself then, that's not going to be me. And so what we found is that for the 140 women, employees of the conservation area, we drew up a chart at one point. How many brothers and sisters did you have? How many children do you have? 12, 3, 10, 2, 5, none. 1 generation, women with economic independence find it very easy to convince their husbands that they would rather have a new outboard motor for their fishing boat than another child. Now, it also helps that Costa Rica has no military. They spend all their money on rural health care and education. They have a higher literacy rate than the United States, even before the last administration came in. All of these folks can read and write. They're all computer literate. They bring technology into their local village, and they have a good time. And they're making a significant contribution to their society. And this is one of the most recent things. Here's Patrona and Callie again. This is a more recent thing. This is a picture that is a certification program. They get their certifications for strengthening sustainable practices in livestock management in the area. So their influence has extended outside of the conservation area into the managed landscapes around the conservation area. So it works. You have to understand the local context. Citizen science, in the sense that Orsay's doing it, that works here, would not work there. But it's very similar kinds of thinking behind it. It's scientists and citizens working together. It's people seeing that things like coping with emerging diseases and coping with the biodiversity crisis is a matter of social service. And then giving people an incentive to participate, which they then find through a very positive feedback is a very satisfying kind of existence. Thank you.